Good evening, all. On behalf of ISHBT, I would like to welcome all delegates on 18th Masterclass. Without wasting any further time, I would like to invite our speaker for today's masterclass, Dr. Mayur Parihar, Senior Consultant, Lab Hematology and Molecular Pathology, Head, Department of Cytogenetics, TMC Kolkata, PI and Clinical Scientist at Tata Translational Cancer Research Center, Kolkata. Over to you, sir.
Yeah, hi, good evening, everybody. So what I'm going to do in next 45 minutes is, so without much delay, I'll just directly go. So what I'm going to do is give you some basics about cytogenetics and how it is applied in hematology, in day-to-day modern hematology practice. I would be dealing more with oncology here, hemato-oncology, rather than talk about the benign hematology here. The slide is not moving. Okay, these are my disclaimers. So when you look at the, let me go through certain basics first. So when you look at the DNA, it just has two jobs. One, it serves as a store of information. It codes for the RNA, transcribes to RNA that codes for proteins, and these proteins are essential for the functions. And the second job it has is it has to ensure that all this information is passed down to the next generation of daughter cells. Now, in order to do that, the DNA needs to have a dynamic structure in order to fulfill both of its functions. So the dynamic structure is such that it is in the form of a thread within the nucleus. And as the cell starts dividing, it is not possible to actually, divide this thread-like material into two equal halves among the dividing cells. So what happens is the DNA starts condensing itself as the cell divides, and then it forms condensed bodies, what are called as chromosomes. Now, these chromosomes are visible only during the mitotic phase, especially during the metaphase. And what is a chromosome is nothing but a molecule of DNA. So when you look at the DNA, as I said, it has different levels of organization. So it can be in the form of a thread, or it can go on condensing to form what is called as a chromosome. And what you see here is that the DNA wounds around what are called as proteins called the histone proteins. Now, this is very essential to know because this is how your epigenetic mechanisms play a role in expression of any gene. So when you look at this part of the DNA, which is in between these histone proteins, that is the DNA which is available for transcription. So in order to control gene expression, the DNA can get wound around these histone proteins when it is not available for transcription, or it can loosen up like a yo-yo, and then it is available for transcription. Now, before I go into further detail, I always love to touch upon this history of the discovery of DNA structure. It's a basically story of four people: Rosalind Franklin, Maurice Wilkins, and of course, Watson and Crick. So at that time, everybody knew that whoever discovers the structure of DNA is going to get the Nobel Prize. And Maurice Wilkins and Rosalind Franklin worked together in the same lab. They hated each other right from the day they met, right? And so what they were actually doing was they were bombarding a DNA molecule with X-ray, and the process is called X-ray diffraction photography or X-ray crystallography. So like how when you put a light on an object, the object gives a shadow, and you can analyze the shadow and know what the object is. So that is what they were doing. They were actually bombarding the DNA molecules with X-ray, then trying to analyze the images that or the shadows that were created. Now, this guy, Maurice Wilkins, was very good with data analysis, and Rosalind Franklin was very good with getting the X-ray images. Sorry. Because it was technically quite challenging to actually get a DNA image, because as I said, DNA has a very dynamic structure. So on the other hand, Watson and Crick, at that point of time, they got along very well together. They thought that the DNA is a triple helical structure. So anyways, to cut the long story short, one day what happened was when Rosalind Franklin was away, Wilkins went to her office, from the drawer, removed this picture of which was called the picture 50, and showed it to Watson and Crick. And the moment they saw this, they realized that it's a double helical structure, and they formed the DNA. So they went and said that they have discovered the secret of life. They went to a pub where they used to visit frequently and made that announcement that we have discovered the secret of life. 3 papers came later that year. But however, only Watson and Crick and Maurice Wilkins got the Nobel Prize for DNA structure. Rosalind Franklin died of cancer because of exposure to the X-ray, and she never got the Nobel Prize. So later, Watson said in one of the meetings that he didn't steal the image; he was actually shown. But what this did was, once the structure of DNA was identified, what you see here is an image from Science where about 150 years of Nature papers have been displayed. So each dot is a paper, a published paper, and how these papers connect with each other. If a paper has actually referred to a work, it's connected to it. And those papers which have been used as a reference lie below it. So you see, before this point where Watson and Crick paper came about the structure of DNA, there were very few papers on genomics. So Watson and Crick referred to a lot of papers in order to describe the structure of DNA. And once that was discovered, there was a huge change in understanding the DNA, understanding the biology, and that in developing technologies. And then you can see there's a fountain of papers that came on genomics. So when you go practically to a lab, what are the genomic techniques that are available? Karyotyping is nothing but you look at all the chromosomes. It is a whole genome of a single cell. You can pick up ploidies, that is gains or losses of chromosomes. You can pick up translocations. You can pick up structural abnormalities. FISH, you use a specific probe. So whatever question you are asking, it will answer only that. You can do single nucleotide polymorphisms, which is like a digital karyotype. It helps you to identify copy number changes, structural abnormalities. And of course, we have high throughput sequencing and focused or targeted sequencing. So when I look at it as a pathologist, I would like to divide these techniques into those that are microscopic based and those which cannot be identified by human eyes. So karyotyping and FISH, which is usually the routine bread and butter of a cytogenetic lab, are a microscopic based technique wherein you use microscope to look at the chromosomes, and you use FISH, and you can actually look at the genes which are using fluorescent probes. And the submicroscopic abnormalities, you have the copy number alterations, which means gains or loss of genomic material. You could do SNP arrays or a targeted screening in the form of MLPA, or you could look at mutations that could be targeted or a whole genome level. Now, you must understand the one basic concept: which lab does what. So a cytogenetic lab studies structural chromosomal abnormalities. It does gene subbinding, FISH arrays, etc. And a molecular lab actually goes deeper and looks at the nucleotides. So it does PCRs and sequencing and so on. Molecular cytogenetics is a marriage between conventional cytogenetic and molecular techniques, wherein you can use probes or sequences of nucleotides to do your cytogenetic study. And the molecular cytogenetic techniques are FISH. I will talk about this in detail as we go ahead. Multicolor FISH, spectral karyotyping, comparative genomic hybridization, and SNP arrays. Now, usually karyotyping and FISH are the routine techniques that are used in a diagnostic laboratory. What is the difference between the two? Karyotype is, as I said, the whole genome of a single cell. You can actually look at all the chromosomes. You can look at the whole genome. You can identify for if there is a clonal evolution, if there are multiple abnormalities. Whereas FISH will only answer the question you are asking. So you use specific probes and you ask a question: is BCR-ABL present or absent? FISH will just give you that information that it is present or absent. It will not say, it will just say positive or negative. It will not give you information as to what is happening at the other part of the genome. So conventional cytogenetics is karyotyping. Usually, for hematological malignancies, we use bone marrow. As I said, chromosomes are visible only during metaphase. And that is why for conventional karyotyping, you need live cells. And hence the samples have to be transported immediately or as soon as possible. They shouldn't be frozen. You need fresh samples here. And what we do is we culture the cells to get the metaphases. You need to understand that in a bone marrow, the cells are spontaneously proliferating. So you do not add a stimulant. You do a spontaneous culture. You just put the bone marrow in a culture media and let it grow, let the cells divide, right? So lymph nodes or solid tumors and in constitutional abnormalities, coronaceae, they have spontaneously proliferating cells. However, if you need to know a constitutional karyotype, you need to stimulate cells. So you take a peripheral blood, you stimulate the peripheral blood lymphocytes by using PHA, which usually stimulates the T lymphocytes, and you get metaphases. If you want to karyotype plasma cells in multiple myeloma or lymphocytes of CLL, you will have to use interleukins and interferons to stimulate the B cells to push them into division to look at their chromosomes or look at their karyotype. So the preferred anticoagulant to be sent for cytogenetics has to be sodium heparin. You do not send a sample in EDTA for karyotyping because EDTA chelates calcium. And then because I said you need cells to be dividing, you need live cells, EDTA will not work. So peripheral blood should come into, as I said, preservative-free sodium heparin. You could also send lymph node biopsies in hematological malignancies. You can do FNACs, and you can send the aspirate. You can send the pleural fluid. We have also fished CSF. And many a times when there is no sample available, you can actually take a stained bone marrow, destain it, and then fish it. So this is what happens in a conventional karyotyping. The marrow is taken, it's put in a RPMI cell culture, put it in an incubator with 5% carbon dioxide. And as the cells start dividing, the metaphase is arrested using Colsumit. If we add some hypertonic solution, we make slides. Those slides are stained or banded. And then we have a microscope with a camera attached. We capture all the metaphases and then analyze them, right? So this is how it goes. I'll just go quickly here. So for peripheral blood, it's a 69-hour culture. Bone marrow, we do 16 to 18 hours culture. Why 16 to 18-hour culture? Because it's based on the cell cycle. If you look at the timing of the cell cycle, it's about 17 hours. So whenever we do bone marrow cultures for hematological malignancies, we culture them for about 17 to 18 hours so that and we add Colsumit there. So every time you're supposed to do 2 cultures, and every time a cell divides, the metaphase will be arrested and you will be able to get metaphases. So as I said, we do 2 cultures. One, we add Colsumit and put it for culture. The other, we add the Colsumit the other day. Now we culture overnight for 16 to 17 hours and then we harvest them. Chromosomes are best studied at metaphase, and the protocols have to be followed because this is what you want to look at. You want to look at the chromosomes that are easily visible, they are well separated, the banding pattern is good, but a small error in following the protocol can result in a clumped metaphase like that. And in these cases, you just cannot analyze the chromosomes. Next question is, what do you mean by bands? So what we do is we stain the chromosomes, right, with a Giemsa stain, and it gives light and dark bands. So a chromosomal band is a part of a chromosome that can be distinguished from adjacent segments by appearing darker or lighter. It's just like the barcode you see in supermarkets. You scan the barcode and you will get your product. Similarly, a chromosome has light and dark bands. Now, you must remember that the light bands have active transcription sites. So the light bands are the ones which will produce your RNA and protein, and the dark bands will not. So the more number of light bands a chromosome has means there are more number of genes that are present on them. So you can see here we have a microscope, we have made a slide, there's a camera attached to be capture the metaphases, and then this is how the metaphases look. These are the interface cells. And then we use a software to analyze, and each chromosome has its own pattern of light and dark bands. And these chromosomes are based on their banding patterns are arranged. Once they are arranged, so it's like morphology, then you look for changes. You look for structural abnormalities, translocations, deletions, and so on. This is what you see, a metaphase from a patient of acute leukemia showing 77 chromosomes, right? So the first part is to actually arrange the chromosomes, and then you start looking for the banding pattern and just see for any subtle change in banding pattern. The moment there is a change in the banding pattern, you know there is an abnormality, and then you try to find out whether it's a translocation, whether it's a structural abnormality, whether it's a duplication, whether it is inversion, and so on. So, for example, this was a patient of AML showing a translocation 816. There can be very subtle abnormalities. These are patients of acute myeloid leukemia showing a 3-3 translocation. This is an MLL gene rearrangement showing 1119. So you see it's a very subtle arrangement. You look at this 19, it shows a dark band here. This is the only clue you will get. So it's a lot of morphology. It's a very laborious process. You need to go by band. You need to identify the abnormalities and you need to pick them up. What is FISH? As I said, FISH is a procedure wherein is a technique wherein you ask specific questions. So you just apply the specific probe. So the first part, what you do is we denature the DNA and then we add the probe. And the probe, what it will do is it will find its place to the complementary sequences of the genes we are interested to study, and we'll go and hybridize, and then we will read the FISH signal patterns under the microscope. For example, 922 or BCR-ABL, the ABL1 gene, you have a red fluorochrome, and for 22, you have a probe which has a green. So normally, in normal cells, you'll have 2 copies of every gene. So you'll see 2 reds and 2 greens. The moment there is a translocation, the 9 will give to 22, the 22 will give to 9, you will see yellow signals. So that is how we look at dual color, dual fusion signal patterns. There are different designs of probes that can be used. There are genes like MLL, which are very promiscuous gene. They have at least 100 different partners in 110 different translocations. So how do you design probes for them? So what we do is we use break apart probe, wherein the green and the red are together in a normal gene, and the moment there is a translocation, they get separated. For example, this is a normal cell. You can see the red and green are together here. This is an interphase cell. What you see here is a metaphase. Here you see the red and green have separated. So these are called the break apart probes. This is for MLL or KMT2A gene. And you can see that this is rearranged. This is a metaphase. You can see this is the normal chromosome 11. This is a 10-11 translocation, and the 2, 3' and 5' end have separated. Now, what do you do when you have to FISH on a solid tumor? You must understand that FISH on solid tumor is technically much more demanding. You need to, there are a lot of processes that go into place. There are pre-analytical factors like the fixation, the kind of wax block that is made, the kind of xylene this is, have you used buffered formalin, and so on. And it's much more technically demanding. We look for a lot of rearrangements on solid tumor. In hematological malignancies, we look for triple hit lymphomas, we look for TP53 gene deletions in lymphomas, we do all kinds of translocations in lymphomas. So these are the pre-analytical recommendations for FISH on solid tumors. Now coming to the basics of parts of a chromosome. So chromosome has a short arm called P arm, a long arm which is called U, Q arm, and a centromere. Now you have metacentric chromosomes, submetacentric, and acrocentric chromosomes. And we do follow an international system of cytogenetic nomenclature, which actually defines how a report has to be given. So whenever we talk about a band, we first write the chromosome number, then the arm symbol, the region number, and the band within that number. So 1P31 indicates chromosome 1, short arm, which is P, region 3, and band 1. We do have banding patterns ideograms, which we follow. When we are doing our reporting, we look at the ideograms and see which bands are involved. And whenever we report in the square brackets, the number of cells that are showing a specific karyotype are mentioned. A lot of short forms have been defined by the ISCN, where DL is deletion, dicentric translocations, isochromosomes, inversions, T is for translocations. So you don't have to go remember all of this, but I'm just saying we do have certain guidelines. So whenever a report is given, we write it in the ISCN format, but we also write the interpretation. Now, what are the types of chromosomal abnormalities? You could have numerical or structural. Numerical are aneuploidies, wherein you can have a loss of a chromosome or a gain of a chromosome. And polyploidy, wherein it's basically just a duplication. So 23 chromosomes becomes 46 chromosomes becomes 69. So multiples of 23 are called polyploidy. And the structural abnormalities include translocations, inversions, insertions, deletions, ring chromosomes, isochromosome, and extrastructural abnormal chromosome. So the examples for aneuploidy are say 45 chromosomes or 47 chromosomes, and polyploidy, as I said, are 69 and 92. Now, let me just go to, skip the slides now. What about the genetic profiling in oncology practice? When do we do genetic profiling in oncology practice? You must understand that some of these tumors, both in hematology and of course in solid tumors, are characterized by their genomic signature. So you can actually identify the abnormality and use that in diagnosis. Or these cytogenetic abnormalities are also used in risk stratification, wherein low risk cytogenetic abnormality patients get a low risk therapy, and those with high risk cytogenetics get a higher therapy. And then, of course, by identifying genomic abnormalities, you have specific drugs like tyrosine kinase inhibitors in Ph positive ALL and so on. In oncology cytogenetics, you need to understand the concept of clone. So, what is a clone? A clone is defined as a cell population derived from a progenitor, and the number of cells that constitute a clone is given in square brackets. So, for example, this is 46XX, translocation 821 in 20 cells. Now, in order to call something abnormal, there should at least be 2 metaphases showing trisomy. To call something monosomy, you need 3 cells or 3 metaphases. To call a structural abnormality, you need 2 cells, right? So many a times I get reports for review across the country, and you see reports like that. Now, whenever you see a report like that, you need to call that lab and ask them, how many cells does the monosomy 7 is present? Because of a technical artifact, if there is only, it can be possible that there can be one cell where the chromosome 7 has got scratched off while cleaning the slide. So you need to ask, is it clonal or is it just a random abnormality? How many metaphases have monosomy 7? So this is the kind of report you're looking for, that 7 metaphases have monosomy 7 and 13 metaphases are normal. So whenever you have one metaphase only showing a monosomy, the clonality is not established. So there you have to suggest to do a FISH, wherein you can do specific probes for chromosome 7 and you can analyze a larger number of cells to prove clonality. Translocation, as I said, is exchange of material between 2 chromosomes. A balanced translocation, there is no gain or loss of material, and unbalanced, there is a loss or gain of genomic material. So what is the effect of a translocation? So a translocation can cause deregulation of genes by juxtaposition to strong enhancers. For example, in the 814 translocation, wherein the IGH enhancers lead to overexpression of the MYC gene in a translocation 814. Or you could have chimeric fusion transcripts. Now, these chimeric fusion transcripts could affect the transcription factors. For example, in 821, the RUNX1, which is a transcription factor, can affect epigenetic modifiers like the KMT2A or the MLL gene. Translocations can affect cytokine receptors like the ALK gene in ALCL, or can affect tyrosine kinases like BCR-ABL1. Let me show you an interesting case. Now, the question is, do we need a blast to diagnose an AML? Yeah, I mean, we need this kind of a blast, of course. This is the kind of blast we don't need, but these are the blasts we see in our hematology labs on a day-to-day basis. So this was a girl, the history was that the whole family had viral fever and everybody had recovered, but her platelets continued to be low even after for at least 2 to 3 weeks after the fever episode. So a marrow was done, and what marrow showed was a lot of hemophagocytosis, as you can see. This is the macrophage, it has eaten a neutrophil here. Now, in a viral, post-viral, you could see these kind of hemophagocytosis, which is reactive. And we looked, there was some amount of dysplasia present, blasts were less than 5%. A flow cytometry was done, and flow cytometry showed some dysplasia, but there was no increase in blast. When we did the karyotyping, we got a translocation 821 with a deletion 19. Now, as all of you know, the WHO right from 2002 has said that if you have an 821 translocation, it is diagnostic of acute myeloid leukemia, even if blasts are less than 20%. Well, our patient had 0 blasts, but we still had to call it an acute myeloid leukemia because the translocation 821 was present. The other abnormalities wherein you can call something an AML is inversion 16 and 1517, irrespective of the blast count. And in the new WHO, a whole lot of genetic abnormalities, recurrent genetic abnormalities, you can call the patient of AML even if the blast is less than 20%. Now, I'll just show you an example of a balanced translocation. This is a translocation 119 seen in patients of acute lymphoblastic leukemia, and what you see here is an isochromosome 9. Right? Now, this was a patient of CML and blast crisis. You can see the patient has a 119 and a 922. So the cytogenetic abnormalities can occur before the blast crisis can present. Right? you can have an unbalanced translocation, like in this patient, you have a derivative 1,7, which is a loss of 7q and a partial trisomy of 1q, and these abnormalities are associated with high risk in myeloid neoplasm. Now, this was a patient of chronic myeloid leukemia who was not responding to treatment. A karyotype was sent to us, and we found 922, an extra Philadelphia chromosome, and trisomy 19, clearly showing that there is clonal evolution and the patient is progressing towards a blast crisis. Yeah. Now you can get three-way translocations. The three-way translocations are similar to the normal translocation. And even by a FISH pattern, you can identify a three-way translocation. What do you mean by inversion? Now, inversion is when a part of a chromosome cuts and then completely turns upon itself. It's called as an inversion. Now, inversions can be difficult to detect on karyotype morphologically. Inversion can be either paracentric, wherein inversion happens on only one side of the centromere, or pericentric, where they happen on both sides of a centromere. So one such example is inversion 16. You can see it's a very subtle abnormality. The co-binding factor beta, which is on the Q arm, MYH11 on the P arm, and whenever the inversion happens, the MYH and CBFB fuse to form what is called to result in a inversion 16 or form a CBFB MYH11 fusion. And this can be easily detected by a break apart FISH probe. Similarly, in inversion 3, you can have the EVI1 and the GATA2 come together, resulting in overexpression, and that is associated with high risk. Insertion, a small part of one chromosome can be inserted to other. Deletion is nothing but a loss of a part of a chromosome. So deletion 5q in myelodysplastic syndromes. An isochromosome is when one arm is lost and the other arm duplicates. So as I said, iso 9q. So what happens is the 9p is lost and the q arm has duplicated. So it's a mirror image of one arm of the chromosome. Patient of CML not responding to therapy showing 922 and isochromosome 17p. So because of the isochromosome 17q, what has happened is the P arm has got lost. The P arm houses the TP53 gene, resulting in deletion of the TP53 gene. Duplications are when a part of a genome duplicates itself. So duplication 1 is seen in, and there's a derivative 19 translocation 119 here. So duplication actually results in additional copies of those specific genes. Now, when you look at karyotyping, you also look at clones and subclones. So if you look at this patient, there are 6 metaphases showing 922, and there are 14 metaphases showing 922 plus trisomy 8. So this clone is now evolving, has acquired trisomy 8, and this patient is moving towards blast crisis. Sometimes what may happen is, this was again a patient of CML who continued to have dysplasia and some amount of pancytopenias. He was on imatinib, and when we did karyotyping, we just found the trisomy 8. We did not find the 922. And so you can have a pH negative clone also come up in patients who are getting imatinib. Complex karyotype is defined as that which has 3 or more abnormalities. The MRC calls it phi. A monosomal karyotype is defined by presence of 2 autosomal monosomy or 1 single autosome monosomy with a structural abnormality. So this was a patient of MDS with deletion 5q on FISH, was on treatment. But when we did a karyotype, it showed a complex karyotype, and this patient soon evolved into an acute leukemia. Now here there's an abnormality of a 3q with monosomy 7. This is again a monosomal karyotype. Let me show you another interesting case, the role of cytogenetics in transplant patients. So this was a patient of CML diagnosed in CMC, underwent a transplant, and he underwent a haploidentical transplant, wherein the XY, so when you do a haploidentical transplant, then the hematopoietic cells, the XY, because a male patient, become XX, and that is when we use FISH. So this is how it would look, XX and XY and XS. So when we did FISH on this patient, what we found was he came back with a headache and there was suspicion of a viral fever. And on follow-up, when we did FISH, because he had undergone transplant, we thought, we were expecting that all cells should be XX, or if he has relapsed, then the cells should be XY. But what we found was, we found about 30% cells showing a single copy of X. And then we asked for a BCR-ABL fusion, which showed additional copies, and the karyotyping showed a deletion of chromosome Y, and that is why we were only seeing a single X chromosome in the fish for XY for transplant. And this is how the probe, and I'm showing you this case so that you should realize that it is very essential to understand your probe designs before you interpret your fish. There was another patient, 32-year-old, doing PhD in US, came to our hospital, got his transplant done. Again, he had a sex mismatch transplant, was okay for 2 years, comes back after 2 years with headache. The molecular chimerism is fine. The bone marrow is, he came with fever and headache. The bone marrow was fine. Immunophenotyping was fine. Now, we were expecting that all his metaphases should have shown an XX, but what happened was that this patient actually was relapsing, and we found 6 metaphases showing XY along with A1122. So even before the disease had come back, even before the myeloid leukemia had come back, we were able to genomically, preemptively identify that the disease is going to come back. So he was then treated actively and aggressively, underwent a second transplant, and is doing fine now. Array CGH is and SNP arrays are used more and more nowadays, wherein on an SNP array you have a lawn of nucleotides on a chip. What you do is you put the patient DNA on it, and you will get signals. Now let me show you one of our patients. Now this was a patient of ALL 51 chromosome, actually relapsed, and we did an array on this. And what we found on our array was that this patient had a deletion 17p, which was not visible on karyotype. So the TP53 gene was lost, and he also had an Icarus deletion, which both of these are high-risk markers, and that is the patient. That is why the patient relapsed. Now, before I move ahead, I just want to show you another case where how cytogenetics can help you to even identify floaters. So this was an 11-year-old boy. Clinically, they thought he's having a testicular case of ALL. And what you see here is you see seminiferous tubule, and you see the relapse. And so this was a case 46. This was the case number 47, which again showed relapse. Now here the doubt was whether this is a floater coming from this patient. Now this is how it looked. It was PAX5 TDT positive. It was a testicular relapse in that case. Now in this patient, we were not sure whether this, because the rest of the testis completely looks normal, whether this piece was coming from the previously biopsy case. So what we did was the one which was clinically testicular had a 1221. The one where we suspected a floater was a high hyperdiploidy. We fished using the ETV6 RUNX1 probe, and we showed that both of these cases were actually relapses. There was no floater problem, and we were able to sort this out. So with this, I have completed my talk for today.